So I don't think I need to introduce to you rare diseases or the EJP-RD because all of you are familiar with that. So I think that the only thing that is relevant here is that the RD-Connect Genome-Phenome platform is one of the systems funded by EJP-RD as a data deposition analysis platform. And I don't know if you can see my mouse, but it's here on the left, together with the European Genome-Phenome Archive, which is mainly an archive, and Decipher, which, as you know, is based in the UK and fulfills a bit of a different function than the RD-Connect GPAP. So these three systems are quite complementary between them, and they have different ways of operating and sharing the data for research or clinical research in most cases. Besides these data deposition analysis platforms, as you know, the virtual platform from EJP-RD, which is basically pillar 2 of EJP-RD, has also many other resources, and they will be explained in this series of webinars that we are doing, and hopefully you also are interested in some of them and will join in later sessions to follow up on them. Next slide. So today we will be talking about the RD-Connect GPAP, and in a nutshell, it is an online system that facilitates collation, sharing, analysis, and interpretation of integrated genome-phenome datasets for rare disease diagnosis and gene discovery. And in many cases, the data that people submit to the system are undiagnosed cases, exomes and genomes from rare disease patients that have been undiagnosed for quite some time. In most cases, all the standard clinical route has been performed on them, and at the end, an exome or a genome is done. And in many cases, even with the genome and the exome, they have not been able to diagnose them. And then when it's shared in the system, that hopefully helps to diagnose this, because it's another way of looking at the data, but other eyes can look at the data in a controlled way, and also data can be shared or discovered beyond the system through discovery mechanisms like matchmaker exchange, connection to Decipher and Phenome Central. And Leslie will talk more about this in a second. So what we do is collect in parallel the phenotypic data and the genomic data, and for the phenotypic data we use standards like HPO, Ordo, and OMIM. And for the genome data, we take the raw data, usually as a FASTQ file or as a BAM file, and then we process the data through a standard analysis pipeline, which is very similar to the GATK best practices. And then we make this integrated data available to authorized users for analysis and collaborative analysis, let's say. This is an early recognized resource for 2 or 3 years already now. And recently we have reached the milestone of over 15,000 genome-phenome data sets in the system. This is not only patients, because there are also relatives which are included in these data sets. The data is pseudonymized, which means that we don't know who the patients are, and the users don't know who the patients are, except for the person that has submitted the data. And this is the person that can go back to the patient and provide any feedback to them if something relevant is identified in the system. Usually when the data is submitted to the system, you have an embargo period of up to 6 months in which the submitter has exclusivity on the data for looking at the data. But we like that the data after that is opened to the rest of authorized users, because in that way, we help, or we aim to help in bridging this gap or reducing this time of diagnosis, which is also the main vision of ERDR, to try to reduce this to less than 1 year. To register, you need to go through a data access committee and sign some papers, and then if they see that you are a bona fide researcher, they will give you access. We log the activity of the users, and you can see which other users have looked at your data, and also the security of the platform is audited and periodically reviewed by an external company. And also very important, here on the left side, you see an arrow that goes to the EGA, the European Genome-Phenome Archive. We collaborate with the EGA for the long-term archival of the data, and for many of the data sets, we submit it also to the EGA. This is always in agreement with the data submitted, so this data also can be archived at the EGA and accessed there in a different way, usually more comprehensively, or at least the raw data, so you can do other types of analysis. So I think that's it from me for now. Just a very quick intro to the system, and now Leslie will go on and do a longer explanation, and we'll go through the demo. I think probably Leslie, you will do an explanation, maybe stop quickly for questions and then do the demo, and hopefully at the end we have some time for discussion. So thanks a lot for joining, and I leave it to you, Leslie.
Yeah, thanks, Alex. So I will first give you, as Sergio was mentioning, an overview of the RD-Connect GPAP, and we'll then have 10-20 minutes for small short demos on specific features, and we'll try to keep questions and discussions at the end. During the presentation, if you have questions, you can also write them in the chat, and here, apart from Sergi, there are also Carlos Garcia and Stephen Lowry from our team that can also start answering them. So to get to share and analyze your data into the platform, you will need to follow several steps. And I will now briefly go into these different steps, and then we'll dedicate more time on the platform itself and to show you the different modules, features, and possibilities it offers to you for analyzing your data. So the first step is that the PI of your group will need to register to the platform by completing a form that is available in our website. It's a 3-step registration process. And as Sergi mentioned before, the requests are then evaluated by a data access committee that will grant you access to the platform or not if there is any issue. Once you are registered in the system, you will then need to submit the phenotypic information of your cases. And for that, to help on the whole process of this, we have developed Phenostore. So Phenostore is a user-friendly tool that will enable us to collect the phenotypic information, but then for you to share and also export that information using standards, ontologies, and control vocabulary. We mainly use, as also Sergi mentioned before, HPOs for symptoms, although for clinical disorders and OMIM for molecular disorders. And we have built the system in a way that it's interoperable. We have APIs that will enable integration with, on one side, the genomic analysis in the GPAP, but also it can be integrated with other systems to facilitate that the data doesn't get siloed in any place. Also, to facilitate for the user the entering of the information in our system, what we have done is to provide disease forms. So we have currently 20 disease forms that are based on the Genomics England data model. And the different European reference networks are mapped to these different disorders. And this was done in collaboration with the Orphanet team in France. And moreover, when Sergi was saying that we also work with specific European reference networks, so when you enter the system, we also have discussions to ensure that everything is in place for you. Otherwise, we can add, as done for several year-end, other type of information, other ways to enter in a structured way all your phenotypic data. So the tool itself, I will show you later. You have different steps that enables you to enter the clinical information, to do deep phenotyping, and also to help for doing differential diagnosis. However, we also enable to do a bulk import of your data if you already have the information in a standard way in our database or in-house systems. So there are these two possibilities. Either you use our user interface or you bulk upload the data through specific templates that we provide. Then once all your phenotypic information is in the system, it's in a structured way, it does not get siloed as we mentioned, and you can also export it using the GH4GH phenopackage. And the phenopackage is a format that has been developed by the Global Alliance for Genomic and Health, and it really enables this secure and standardized phenotypic data portability. And many tools are now built for analysis, for example, to intake the phenotypic information through this format. So this is really a way that we all get and exchange the phenotypic information in the same manner. So once the patient information is in the system, you will then need to submit the corresponding genomic data. And for that, there is like a two-step process. One is to tell us or give us the metadata of your experiment. For example, if it's an exome, a genome, the kit that you have used, etc. And you will then send us the files, the FASTQ files, as Sergio was saying, or in some cases we can also accept the BAM files, raw BAM files, that you send us to our server, and then we will process the data through the analysis pipeline. So we use a standard analysis pipelines to process the data. We make it available for analysis then in the system. You will have access to all your phenotypic information in Phenostore, as I previously showed. Then this module is connected to your genomic data, and we'll see later some example. It will help you for data filtering and also interpretation and prioritization of variants. And it's also connected to other applications, so that the cohort application that we'll also see later on, that will enable you to create on-the-fly cohort based on phenotypic traits from your patients, but also from other patients in the system. And this is very useful, for example, for gene discovery. You will also be connected if you have granted access for those patients to matchmaker exchange, and then this will help to connect with databases such as Decipher or Phenocentral for patient matching. I will also show you now a couple of examples for that. So here is a screenshot of the Genomic Platform, where you will be able to analyze, filter, and interpret your data. I will show you some of its main features afterwards, but basically the platform will enable you to filter your genomic data using the typical annotation, also to create on-the-fly gen list based on phenotypic information, disease information inside our system, but also we query other resources such as Panel App, and it gives you the possibility to use this curated panel at that specific moment. We have tools such as Exomizer for variant prioritization that are integrated in the system, links to multiple external resources at the gene or the variant level. We'll see that later. And then we have several functionalities that will help this collaborative environment and the search across all the data that is in RD-Connect, or the data discovery using Beacon or Matchmaker Exchange. So just to mention that the platform is using in several European projects for sharing, analyzing, and for data discoverability. And here's just an example with SolveRD, which is a European funded project which aims to solve the unsolved rare disease cases in Europe, and in collaboration with several European reference network. And now we are collecting and analyzing data from the goal is to reach 19,000 unsolved cases, and we have also already done half of that. So we are. So apart from the regular case-by-case analysis that we'll see also a little bit in the demo, you can search for specific genes of interest across the more than 57 samples that are available in the system. And this feature is, for example, very useful when you have a candidate genes to quickly see if there is any other case that has variant, truncating variants, for example, in that same gene. So you would have this information very quickly, and then you can directly from the platform contact the submitter of that sample and start a collaboration outside our system, and then it follows you to continue with that. As mentioned, we are part of Matchmaker Exchange initiative for anonymized data discovery. So the system will enable you to know if there is a patient with a similar phenotype and genotype to yours in any other of the databases connected to this network. And here's just a screenshot on our platform. So on the left, it's the form from one of the cases that you might have entered in the system. We automatically retrieve all this information, mainly on the phenotypic and the genomic information. We submit this information to the Matchmaker Exchange network, and then you retrieve a list of cases with a score, a similarity score with other patients in the system. And here you also have the possibility, you can see on the right of the screen, to directly contact the submitter of that sample and start a collaboration based on these findings. And finally, before now moving to the demo of several features, I would like just to acknowledge the team. So the team is led by Sergi Beltran, and there are several engineers and informaticians behind the development of the platform, which is led by David Epicia. And I also like to mention the people involved in all the data analysis, mainly Stephen Lowry, Marcos Fernandez, and also Carlos Garcia, who is in charge of the logistics. So now I will move to the demo, or maybe Sergi, should we have, if there are some questions, general questions, and then I can go for the demo. So we have also a little break.
Let's take a couple questions. We had two on the chat that we answered regarding the need to transfer data between CPMS and the GPAP. As I said in the chat, we use Phenostore both for input and Phenopackets for both input and output, among other formats. But I don't remember now if CPMS is already compatible with Phenopackets or not. But we can take other questions. I don't know, Katerina, can people speak up, or do they need to raise the hand?
Normally they can just unmute and speak.
Okay, so any questions from anyone? Okay. Is REDCap for data available as well? Could I ask for help in establishing connections, e.g. email or frontal cross? Nina, yeah, no, we don't use REDCap ourselves, but I'm sure that there are ways of transferring data to and from there. And yes, if you put us in contact with them, we can discuss about this. Any other questions? Okay, so Leslie, I guess you can go on with the demo now. Okay. You will need to zoom in a little bit so that we can see your screen. Otherwise, the text is very small.
Okay, yeah, I will just try. I will share from my other screen, which is smaller and should be better. Let's see if it works. Is it better like this?
Yeah, it's a bit better, yeah.
Okay. Okay, so this is the platform landing page, so you can access it through this link, and I'm sure we'll share the slides afterwards, and you will have this link too. Just very briefly, so that you quickly see where you can access the different parts. So here you would have access to the registration form to get registered to the platform. Then once you are registered, you can access the Phenostore module here to access phenotypic data submission, and you have guidelines always associated to the module so that you can learn how to do the different steps. Then you will access the submission of the experiment and the files metadata, the same here with the guidelines, and on the right is where you will have access to the platform. And you also have video tutorials and guidelines to help you on that. So I will start by showing you the genome analysis platform. As I have a zoom in, letters and so might be a little bit not very well configured, but when you do it in your computer, it should be okay for that. So when you start entering the system or entering the platform, there are three main parts. The first one is here on the top and will be which samples you would like to analyze, the case you would like to analyze. So in this example, I have three different samples, so that would be a trio. You enter the IDs of each of your cases. And then at the bottom of the page, you will have a summary of the phenotypic information of the cases, and also with the HPO terms entered and the clinical information that are in the system, so that you can quickly have an overview of that. Then the next step will be to define the filters you would like to apply to the genomic data. Today we don't have time to enter into all this, but we have a demo tool that we can send the link so you check all the annotations we have. The idea is that from there you will select which filters you would like to apply to your genomic data. I will only show you one specific one, and then at the bottom of the page you will get, once you apply your filters, the list of variants that meet that criteria. And from here, so this is the results section, you will have a different annotation. When you select a variant in the results, in this medium bar, you will have also deep annotations or many annotations at the variant level, so that it can help for the interpretation of that possible variant. Also to mention that you have links to many external databases at the gene level or also at the position level, so it helps you to go either at that specific gene or that specific position in many of the most useful databases for interpreting the data. And just as an example, if you would like to apply the ICMG criteria to define the clinical significance of your variant, you could go to link directly to Barsom, and then you will have the information of that variant in the system, and you will be able to play with the different criteria to define the pathogenicity of your variant. To further filter the data, as I mentioned, there is a set of filters that I won't enter into too many details. I would just like you to see the possibilities you have and how we connect the genomic data with the phenotypic information. I have previous filters that I delete. So we have different ways to create on-the-fly gene lists so that you can filter your genomic data. For example, here you would be able to input the HPOs that from that specific case that you have entered in the system. So here I have the list of HPOs from my patient, and then create a gene list based on the HPO ontology, for example, or other resources such as Mendelian or RD-Connect. In the platform, also, if you hover the mouse in the different places, you will have more information on what's possible for that. As I mentioned previously, and it's also quite useful, is the connection with the panel app. So you can type your disorders, and if it's connected to any type of panel in panel app, you can also retrieve all the genes that have been curated and associated to that panel from Genomic England. I would also like to mention that once you are in the results section, so you can either filter your data, have few candidate variants and look into it, but we also provide a variant prioritization tool, which is called Exomizer. So directly from here, we input again the HPO, so the phenotypic information, and what the Exomizer does is that it will rank your, in this case, these 52 variants according to a score to see which one is more likely to be disease causing. So you would just need to submit this and run the analysis, and then you will get your results. And if I go here to the prioritize genes, what I get is for each of my variants, as I mentioned, this Exomizer score, which ranges from 0 to 1, and 1 being likely pathogenic. Of course, these are based on prediction, so you always need to check the data. But in this case, for example, in the case I'm analyzing, this would be the causative variant, and indeed it has a really good phenotypic match and variant pathogenicity match too. So these are some of the key features that you can identify in the platform. Now I would like to also briefly show you the other modules. So when you are at the sample level, as we mentioned, everything is connected between Phenostore and the Genomic Platform. So I could either look at the phenotypic data as we have done before, a summary of what I have, but I can also click on here and directly go to my form, my case all the information I have entered in the system in Phenostore. So now I'm redirecting you to this case in Phenostore, how it looks like after we have entered the information. So you have the summary of basic information of your case, the pedigree. Then you have the information on the symptoms entering the system. We use HPOs, and we also enable to add the severity of the symptoms with a timestamp so that you can follow up on your patients. There is family information, medical history, also the possibility to add the measurements to see where your patient stands comparing to the others. The diagnosis, as we mentioned, we use Orphanet and the Omni ontologies. You can also define if it's a suspected diagnosis or a confirmed. In this case, it would be a confirmed. And another feature that the system enables you is to enter the genetic information from previous testings. For example, in this case, there is the gene DOC7 that was screened and was negative through an NGS panel. But then another gene, RARE1, which is the one we identify the variant in exomiser, let's say, has been defined as causative by whole exome sequencing. And here you can also have information on the specific disease-causing variant. So this is at the sample level, your case level. From here, you can export this information as a PDF. You can always always edit that information and have access again to the form to, for example, add other symptoms that might have appeared in the system. You can export your data, as we mentioned, using Phenopackage also. But apart from that, apart of having the information of your specific case, what the system enables is to have an overview of all your data, so to browse all your cases. So here you have access of all the information you have entered in the system or has been specifically shared with you in the system. You have an overview of the data. You have several graphs that show you the number, for example, of unsolved and unsolved cases or the affected status of the data you have in the system. You can also filter your data and export this information. You can export in an Excel format, and also, as mentioned, you can export all your data into Phenopackage. So it's really enable interoperability within systems. Okay, and then, so here we said Phenostore is to enter the clinical information. We link it in the Genomic Platform, but then we also provide a module for creating cohorts. So this is the cohort application. And here you will be able to browse your data, but also all the data that is post-embargo, so that is available for the authorized user in the platform, so that you can create specific cohorts based on, as you will see, clinical information or experiment information, for example, according to a project that I would like to analyze, or only for samples that belong to a specific ERN, so I could filter for that, or by any of the characteristics that are included in our system. So by all the characteristics that are available in Phenostore, mostly all are also available here to create your cohorts. We also enable from here, I'm switching tabs so that I don't lose time doing the queries, but you can enter any of the symptoms of interest. For example, in this case, I have entered intellectual disability, so I have filtered by all the patients that have this HPO. And now I can see the other HPO associated to the symptoms and keep filtering my data if I would like to create cohorts based on that. But I can also do it based on the order, so look for a specific disease and only retrieve the patients that have this order in the system, and the same with the OMIN for the molecular diagnosis. all these searches are possible with an or an add addition, so that you can really play a lot with the clinical data and create your cohort. Once you have defined your cohort, then you have a table with all the information that you can also export to further analyze it, or send to the GPAP so that you can make an analysis using the samples that you have defined and the experiment that you have defined in this specific cohort application. So this is what I would like to show you next, is how to use all the data that is in the system. So apart from doing the case-by-case analysis, we also enable you to query all the data that is in the system. So I'm doing a search across all. For example, here I would have accessible more than 14,000 samples. But as I mentioned, when you create your cohort, I could also select to do this analysis only on my cohort of interest. So here I have preset the system to show you an analysis for all the samples in the GPAP for a specific gene of interest. So the gene is the GCDH. I have run this analysis for variants that have a high impact on the gene in the system. I retrieve 19 variants belonging to more than 300 experiments. So this is like the aggregated information, and then on the right I have the list of these 19 variants. I can also see how many samples each variant has. So I can, for example, discard this one if I'm the one that has more than 300 experiments associated. And from here I can then look at the clinical data of the experiments that have those variants, and here I have a summary of that data. I will also have the order if entering the system, the HPOs, and more importantly, if one of these patients is of interest and has a variant in the gene I'm looking for, I can click here on contact, and this will send an email to the data submitter to start a possible collaboration. Yeah, I think this is pretty much what I wanted to show you. Yeah, there are other things that maybe might come up if you have more specific questions. Yeah, but let's open to questions and see if we can show other features.
Okay, thanks, Leslie. We showed a lot of things, so I don't know if people got lost on the way. Let's see if someone has any question. Otherwise, maybe you can, let's wait for a minute or so. Otherwise, maybe you can just show one example of a case from top to bottom, how you would do it, and then we'll maybe see all these functionalities at work. I don't remember if Leslie mentioned that, but we do have a playground, which is a system that you can use at any time that you want to go and test the system, so that there are a few examples, there are a few cases, and we provide you with the phenotypes, and then you can try to find the causative variant in the system, so you will be able to enter the case, the father, the mother, and then find out what is the inheritance that you think it should apply, and then apply different filters to try to solve the cases.
Yeah. So if there are no questions, maybe I can do this and show you briefly a case-by-case application.
Yeah. So Nancy's asking us if all the BCF data are generated with the same pipeline and the reference genome, and the answer is yes. And then she answers if that means that then you would need to update across the board from time to time, and the answer is yes again. We are indeed now, so all the data it's now done with version 37 of the genome, and if I'm not mistaken, GATK 3.6. And now we are running all the data with version 38 and GATK 4 point something. So yes, we are rerunning all the data. What we do update though is that, more often than that is the notations of the data and the databases in the system. So even though the calling is done maybe some time ago, then the notation is still updated. Okay, so Leslie, maybe go ahead and do one case.
Okay. So yes, as I mentioned,
we have 20 minutes, so don't need to run.
Okay. So as I mentioned, well, here I'm using the playground instance. So we have genomes, public genomes, where we have inserted a causative variant. So it's public data here. So what I would do first is to select the samples I would like to analyze. So let's say I would like to analyze a trio. So here I'm selecting case 1. As I mentioned before, once you select the patient to analyze, at the bottom of the page, you have a summary of the clinical information for that case. So here we have a male that is affected. We can see from the pedigree and also the information that we have in the system that it's a sporadic case, so we cannot clearly define the inheritance. Sorry. And then we see that the clinical disorder that is suspected is a congenital myopathy. And these are the symptoms associated that have been entered in the system using the HPO terminology. So basically, we have muscular hypotonia, then skeletal muscle atrophy. So it's really a neuromuscular disorder. And we also have information on the negative HPO, so that the patient does not present intellectual disability. So in this case, we also have information from the parents, so we can do the analysis with a trio approach. So we'll be entering also the samples from the mother, so 1M, and from the father. And once we have our trio then put in the system, the next thing that we will have to do is to define, according to the suspected inheritance, the genotypes of the different individuals. In this platform, it's very flexible, so you can really play with the different genotypes that you would like to assess. As a first try, for example, here we could look for de novo variants. So I would select my case, so my patient, as ref-alt, meaning that the variant is heterozygous for the alternative allele. And then in both parents, I would select that both are homozygous for the reference allele. So I would select the ref and unselect this ref-alt option. Then here on the right, you can also play with all the quality metrics that we define. We have some default set parameters, but you can modify them. And also, when available in the system, you can also add the CMV analysis on top of it. So now here in the middle, we have all the different filterings. Again, I won't enter into details of all of those, but for example, we have one that is broadly used, the variant class, according to SNP. So here to define, to try to identify variants that might have a high impact at the protein level, so meaning that disrupt the protein, such as nonsense, frameshift, or variants that affect the splicing. Or we can also select for variants that have a moderate impact, generally meaning that it's a change in the amino acid, so non-synonymous substitutions. In all the platform, as I mentioned before, if you hover the mouse, you will have this description of each of the filters, so you know what you are applying. We also have links, for example, for the ClinVar classification, where you could directly search for variants that have been reported as pathogenic or likely pathogenic in the database. Then one of the other filters that we usually apply is the population frequency, and we use mainly Nomad, but also RD-Connect enables the internal frequency, so you can also use the information from all the datasets that are in the system, and this is really useful, for example, for getting rid of artifacts from informatics pipeline or for a more common variant in specific population. So in this case, I could, as we are on rare diseases, I would search for variants that are present in less than 1% of the population according to the Nomad database. So if I run this analysis, so now I am looking for de novo high moderate variants that are rare in the population. Only by querying, doing this query, I retrieve the 52 variants. This is the case I had before. So 52 de novo variants, and from here, as mentioned, I could either use the Exomizer as I show you, or look into these variants. But to really try to reduce this number, what we usually do is to try to apply more specific filters based on the clinical information. And indeed, for example, if I take the HPOs that have been entered in the system from the patient, and I create a gene list based on this information, so for each of the symptoms, I will retrieve the genes that have been already associated in this database for those. Now I can filter by the gene list and try to reduce my number of variants. So if I rerun the query, now applying the HPO gene associated, I reduce my number of variants to 6 variants. So from here, it's really a number of variants that can be humanly manageable. So I could really look into all of those. What I have not shown before, but is also of interest here, as I'm using the playground instance, it's not working, but when I select one of these variants, apart from the information I have here on the top, on this right part, you have a link out to the IGV position, so to the alignments of that band. So here I'm not in the main platform, so you don't have them, but for the SolveRD datasets, we link to the EGA and to those alignments, and what you would retrieve is this type of information where you have the IGV browser, your specific variant, and you can see the different alignments and where it is. And this is very helpful to confirm that the variant is real or not. So from here, we could do what we've done before, the way to Exomizer, that will prioritize the real one variant. So the next step would be to check this real one variant. And indeed, we can see that here in the candidate, there is a number, which means that this variant has already been identified in the system. And the corresponding information is here on the top at the candidate tab, where if someone else from the RD-Connect platform has tagged a variant in that specific position, you will see the information here. So here we can see that in these samples, there is they have tagged this variant associated to an autosomal dominant inheritance, and the clinical significance that has been attributed is pathogenic. So this is really helpful, so that to help you on the interpretation of your own data. And if you want to also tag that variant or any other variant, you just need to click here on the tag functionality. Here it tells you that this tag will be available to the RD-Connect users, and you will have this form that is pre-filled with the main information. You will just need to enter the additional information that is not knowable, computer knowable. So, for example, the mode of inheritance associated to your patient and to your pedigree, and the clinical significance that you have attributed for that variant. And then you would submit this tag, and it will be added to the database. This enables you to record the causative information. This will be linked with your Phenostore entry. You will have the variant information there. And it's also helpful at the community level, as it helps to either identify a causative variant, or you can also tag the benign and likely benign variant that you have spent time on doing the interpretation, and you can also quickly discard them afterwards. And so once you have identified, tagged your causative variant in the system, what you can also do is to share all this query and this analysis to your colleagues, for example, so that they can directly look at this, at the search you have done and at your results. And for that, you would use the share functionality here on the top that will give you a link that you can copy paste to your browser, and you will be automatically redirected to this same query, so that you can also retrieve this 6 variant or keep it for record or share it with your colleagues.